So today we are just looking to revise a bit about the digestive system. So we've looked through all of the different organs. We've understood that digestion is all about breaking down big pieces of food into small pieces of food. And we divided our digestion into physical digestion, that's the big chunks of food that we take in and how they're bitten and they're grinded and ground down and churned about inside the digestive system to get them into smaller pieces which increase the surface area for the chemical digestion to occur. Chemical digestion involves predominantly enzymes, but also there's a bit of bile involved in there too. So we've made a list of all the different organs that you need to know about, plus some special features about each of them. Now I'm going to start off at the top of the digestive system with the mouth. I'm going to refer back over to the board, which is where we have some physical digestion, the churning with the teeth and the tongue, positioning that bolus of food into the right place so it can be ground down into small pieces and then swallowed. We also talked about the idea that there's amylase, which is an enzyme, and we're thinking of enzymes being a bit like Pac-Man, but at a molecular level, chopping up big pieces of food into smaller pieces of food. I'm just looking around for my model, I'll show you that in a minute. So our food is then swallowed down through here. So just remove the heart for a second and look behind the heart. Now this tube at the front, you've got two tubes running down our food pipe. The esophagus is the next part, and it's a bit like a muscular wall, a muscular hose if you like, which moves in a wave motion, pushing food down towards the stomach. And if you stood on your head, don't try it, but if you did, try to drink a glass of water, that peristalsis movement, that's what we call it, would actually push water against gravity uphill towards your stomach. Don't try it. Let's take the diaphragm out. Below the diaphragm, our food is now coming into this part here, which is the stomach. Stomach does both physical and chemical digestion. So down here, we've got physical, that churn is basically a muscular sac. Let's look at it a bit more closely. So it's a muscular sac, and inside a very special lining which protects it from the acid and from the pepsin enzyme which is in there. Of course, the acid is produced in the stomach walls in order to help pepsin work at very low pH, very acidic pHs, which is where that enzyme works best. That digests, it like pre-digests proteins from long proteins into smaller chains which we call peptides, and eventually they will be digested into small units called amino acids. So we've got a bit of physical and a bit of chemical digestion going on in the stomach, and it's going to take about a couple of hours for all that stuff to churn about and get all that pre-digestion of proteins done in there. Now don't forget the amylase is still working in here on starch, chopping up those long starch molecules into individual sugar molecules while that pre-digestion of proteins is taking place. Then we move on after a couple of hours, and the stomach releases the contents into this next part. The next part here is going to be called the small intestine. The moment it enters the small intestine, we're just going to take part of the large intestine away. Small intestine's narrower, but it's a lot longer. Small intestine, think it's smaller because it's narrower. The reason it's narrower is to increase the surface area for absorption of all the products of digestion. Now, of course, before that absorption takes place in the small intestine, the rest of digestion has to take place too. That means all three families of enzymes, which we've listed over here. In the small intestine, we have amylase, which is a type of carbohydrase enzyme, the A's meaning enzyme, and this is breaking down the carbohydrate starch. Being a long molecule, it's a complex carbohydrate, it's going to be chopping that up or digesting it into small simple sugars, simple carbohydrates, sugars. That's amylase, so one family. The second family, protease. We've got more enzymes being produced in the stomach. We've got trypsin in there too. We've got pepsin and trypsin working. They're both protease enzymes, digesting those molecules of proteins into tiny amino acid links. Imagine like a link in a chain. You've got all these different links in that chain. For starch, big long chain, each link is sugar, a glucose molecule if you like, and for proteins, each link in the protein chain is called an amino acid. Now we also have lipase. Lipase is a family of enzymes that work on fats, and the proper word for fats are lipids. That's one of the proper words for fats. And if you've ever heard of liposuction, that is the sucking of lipids out of the body, this removal of fat from the body. So that's a good way to remember what lipids means and why lipase is relevant. Of course, all that chopping up's happened, And as that chopping up has happened in the small intestine, we then have this marvellous structure inside, which we have in the background, which is our small intestine with its folded walls to increase the surface area for absorption of nutrients. If you look at any one of those folds, you'll find that there are lots and lots of these villi. So we have one villus or lots of villi. There's loads and loads, millions of villi. These are finger-like projections that stick out into the lumen of the small intestine. And what that does is increases the surface area for absorption of these digested molecules of food that you've taken in your diet. Inside each of these villi is a very, very high surface area capillary network, which is designed to then be so as close as possible to the digested food in the lumen, so it can diffuse through the wall of the villi, through the wall of the capillaries, and into the blood plasma, where it can be carried off and used around the body, or stored for later use. If you look at villi, even the very outer layer of villi have microvilli on the surface, further increasing the surface area where you can absorb these nutrients that we're spending such a lot of energy in the body trying to digest in our food. Okay, this is why you get tired after a meal. Lots of oxygen is diverted to the small intestine because it really does get busy during that digestion process. Now, the moment we get into the food gets into the small intestine, the pancreas and the liver both start doing their jobs. So let's pop the liver in place now, and our liver is going to be secreting some bile, which is a yellow, smelly, yucky tasting substance from the gallbladder here, and that's going to be secreted into the top part of the small intestine. The reason it's there is to think of it a bit like washing up liquid. It's great as like washing up liquid in that it digests big droplets of fat into very small droplets of fat. A bit like when you put washing up liquid on a frying pan after you've used it, and those big blobs of fat turn into very, very tiny blobs of fat, so it's much easier to wash away. Similar process here, but once those big droplets of fat in the small intestine are dissolved, emulsified is the proper word for it, then that emulsion can be attacked by the lipase enzymes so much more quickly because of that high surface area. So this high surface area keeps cropping up, and there are lots of parts to the digestive system which help that. So that's the liver. Now the bile also neutralises the acid which has been produced in the stomach, because that acid is going to cause you massive damage in the small intestine. That wall's not designed like the stomach wall. It's going to get really quite damaged by the acid, so that has to be neutralised. In fact, it's more than neutralised, it does become slightly alkaline in the small intestine. Okay, and that aids the digestion process. Now we've got this leaf-shaped organ here also feeding in, and don't forget we've got the liver over here, we've got the pancreas, so food is not actually moving through either of these organs, but they are involved in the chemical digestion process. Our pancreas is feeding right in the top here of the small intestine, and it's putting the gastric juices in that are needed. It's helping the small intestine out. There's amylase, there's lipase, there's protease enzymes, all three families being produced in the pancreas and secreted into the small intestine. So if you're ever asked, where are all three of these enzyme families found, you will be able to answer the pancreas and the small intestine. And by default, if you're ever asked about any of the three families of enzymes, protease, carbohydrates, or lipase, you can always put the small intestine and the pancreas down as an answer as to where you would find it in the digestive system. Okay? So next, we've got those talked about. Next thing is looking at the large intestine. Now, I took part of the large intestine off just a minute ago. Large intestine is going to be fed into through the bottom here, and we've got the little appendix here, pretty useless, doesn't do anything in the modern digestive system. But the large intestine at this point has, just lost the lung, has the waste basically that the body doesn't want. The body is not chosen to absorb. The stuff you take in your food which cannot pass through the bloodstream, through the small intestine wall into the bloodstream. So you've got your waste plus all of those juices. We've got loads and loads of water in here. Now if your large intestine doesn't do its job, you end up with diarrhea. And that gives us an idea about what the job of the large intestine is. It is to remove and absorb as much of that water as possible from from the waste. Not too much, because we still need to be able to pass faeces out of the body. Otherwise we'd end up constipated, and that in itself would cause problems. So it absorbs as much water as possible from the waste, and then of course the food is then ejected through the anus at the end of the process. So let's just have a look at that digestion of amylase, using amylase, that digestion of starch into sugars, and remind ourselves of what we did with the demonstration earlier today. Okay, so here what we did was we set up some model small intestines, and our model small intestines are made from visking tubing, which is similar in a way to the small intestine in that it has tiny holes in it, so it's semi-permeable. It only allowed very, very small soluble particles, in other words the products of digestion, to actually fit through. We filled this one full of starch solution, so just starch molecules in there, and we decided that we did not expect the starch molecules, being big long chains, a bit like this, to be able to fit through these tiny little holes in the visking tubing wall. And then what we did was we filled it full of blood. Okay, so our water is representing the blood that surrounds, or that comes into very close contact with the wall of the small intestine. That's where the nutrients are supposed to be absorbing through the wall into our blood. So we took some samples of our blood and we tested it with iodine. The iodine here would go black if there was any starch present. No starch present in the blood itself, so no black colour here at all. We also tested it for the presence of sugars, just in case, for whatever reason, we thought, or we'd found any sugars actually being produced inside the small intestine and then making its way through. We thought that wouldn't happen because there's no enzyme in here, it's just starch. So there shouldn't have been any coming through because there wasn't any sugar being made. That's Benedict's before, and this is Benedict's solution afterwards. Now it'll go yellow if there's a little bit of sugar, and we've heated it in this water bath. It'll go green if there's a bit more sugar, and it'll go a red colour if there's even more sugar. There's not been any change there. So, exactly what we expected. Starch cannot fit through the wall of the small intestine, using our model here. And without the enzyme, amylase, there's no digestion happening of the starch. Now this has actually been going now for about 3 hours since the lesson earlier today. Let's have a little look at the other one, because in this other model intestine, we put starch solution and we put amylase in it. And we tried doing the same thing. So we tested for starch in the blood on the outside. Did we think starch was going to make it through? No, because the holes in the membrane are too small. Starch is too long. It's this great big molecule here. So let's test that, the outside, and we did that, and that was the result. We didn't get that black colour that we saw in the lesson. Iodine did nothing. So there's no starch that can actually make it through the wall of the intestine. What we also did was we thought, well, what's going to happen inside this tube, inside the visking tubing? We thought along would come Pac-Man, this is our amylase, and actually snip each of these bonds here and release sugars individually over time, and that those sugars would be a lot smaller than the original starch molecule. So we'd end up with lots of individual sugar molecules, which we thought could fit through the wall and might diffuse from the high concentration inside the tube to the lower concentration of sugar in the blood outside of the tube, and tested again to see whether the Benedict solution would change colour. During the lesson we set up this tube here, and I don't know if you can see from where you are, this was after about 15 minutes. There's a slight sort of yellow tinge at the front there, so we got a little bit of evidence of some sugars having been produced, not only produced, but actually make their way through the wall. And this one here, far more convincing. This I did just half an hour ago, so I've left it in here, working its magic in the water bath. We've got a clear colour change here. There's been plenty of sugar produced, not just produced in the small intestine here, but it's diffused its way through into the blood outside. So there's a summary of organs in the digestive system. You'll need to be able to recall what the names of the organs are, label them on a diagram, be able to explain what their function is and say what their special features are that allow them to do their job. You're going to also need to know about the chemical reactions for all three types of enzyme family. So that's something you might want to look up. I've covered the amylase with you, so we've got starch turning into sugars, but you'll need to look up proteins into amino acids and your lipids into glycerol and fatty acids.